Programming biology without losing control. In the last episode, we saw how molecular logic molecules could one day act like tiny circuit boards inside our cells, sensing, deciding, and regulating our genes. But there's a catch. These modules can't be fully tested or programmed in a dish. Real control has to happen inside the living body, and every human body is different. Each person needs their own adaptive program, one that responds to changing environments, diets, and daily activity, all built on top of their unique genetic blueprint. The starting point is different for everyone, but the end goal is the same: a stable, long-lived system. We all know that's a hard problem. Could one wrong adjustment cause the system to spiral out of control? Genes reshape their own environment. Gene activity never happens in isolation. The expression of one gene depends on many others, and its effects ripple outward through the system. When a gene turns on, it doesn't just use shared resources. It can reshape them. Some genes build proteins that draw energy and materials, while others create new resources by converting one compound into another. Each reaction taps into the same chemical economy that every other cell depends on. As these local changes spread, they begin to influence distant pathways and global signaling networks. For simplicity, we can think of these interactions as coupling forces: shared resources, regulatory overlap, and systemic signaling. Together, they connect every change to everything else, making even a single adjustment ripple through the system. Tuning the picture of life. Imagine an old television set from the 1970s, the kind with knobs to adjust the picture. Each knob controls part of the signal, but turning one shifts the others slightly. You keep adjusting back and forth a few times until you get the best picture. Now imagine each knob as a synthetic controller for one gene inside the body, turning expression up or down. And instead of 3 knobs, there are thousands. In the last slide, we saw how one gene's output can reshape its environment. But inside the body, thousands of these synthetic controllers may be active at once, all interconnected. Turning one changes the balance for others, round and round. With that old TV, you could see the picture as you tuned and know if it was getting better or worse. But right now, we can't see what our genes are doing in real time. Move one control too far, and another signal can fade, or loop back in strange ways. Before we can safely program biology, we have to understand how information flows through these networks, and how risky it is to start tuning in the dark. A stable network at 10% coupling. To understand the risk of programming genes when we can't directly see the effects of our adjustments, we need a simple model of what might be happening behind the scenes. In this model, we start by changing one gene up or down, adjusting its normal output through a logic circuit controller. Each gene can influence about 5 others, but only by about 10% of that initial adjustment. That's step 1. Each of those 5 genes then affects 5 more, but now at 10% of 10%, just 1% of the starting signal. By the third step, the effect has dropped to a tiny fraction of a percent, barely measurable. Even if there is a feedback loop somewhere in the network, it's too weak to cause real disruption. That is what stable coupling looks like: local changes that ripple outward, fade quickly, and leave the system balanced. An unstable network at 30% coupling. Now let's turn up the coupling strength. Suppose each gene still influences about 5 others, but instead of 10%, the effect is 20 or 30% of the original adjustment. The ripples no longer fade as quickly. Most of the signal still weakens with distance, but some paths loop back before they die out. Those returning signals overlap and reinforce one another, keeping parts of the network oscillating instead of settling. Feedback loops that were harmless at 10% coupling can now become self-reinforcing. Each cycle repropagates more of the signal, creating runaway loops that can push the system past a failure point. This is what unstable coupling looks like. A small change made with good intentions can cascade into chaos if the links between genes are too strong or too many. What is the next step? In this series, we've been exploring how molecular logic circuits could one day reprogram our genes from the inside out. But there's a catch. We don't yet have a detailed map of the network we're trying to control. Even small genetic adjustments can ripple through that network and trigger failure points. To solve this, we'll have to think outside the box. The key is visibility, even if it comes through indirect detection. If we could watch those ripples at the molecular level, we could learn how gene behavior modifications propagate in a living person. And by applying the principles of information systems, we could spread out the risk, learn from micro changes, and map the body's gene coupling network. In the next episode, we'll look at how molecular biosensors could make that possible. Because we're only one small step away from the next revolution in biotech.